Okay, before we get started, just a little shout out. Micro Club is trying to get started again after being off all last year. So if you're interested in that, that meeting is tonight. Apparently, if you go in through Laker link and join the club, you can get the Zoom link. That would be easier. So just a little shout out there. Remarkably, everybody's taken the exam. So I don't think even in a non-COVID year, I'd ever had everybody all have makeups done by 2 days after the exam. So this is what the class statistics looked like after the curve. So I curved it to about 74%. So the average was 74%. The way that I curved that, I took the number that you got wrong and I multiplied by 0.9 instead of 1. So it's a little creative way of curving actually. The more you miss, the more it helps you actually. And then I added a couple points on top of that. So before the curve, the mean was a 68, brought the class average up to 74. That was a little bit lower than what I hoped, but that's where we're at. Not necessarily, you know, the ideal distribution that I would hope for, but this is where we start from. Lots of time to improve. Some of you have been into my office already to look at the questions that you missed. I would encourage you to do that. I know it's hard. There's a lot of you and my office is small, but do try to come in and look over your exam. I do want to go over a few questions. So these were the ones that were most commonly missed. Okay, and you know, if you do have comments about any question, that helps me to write better exams next time. If there are words in the question that confused you, it's helpful that I know that. Occasionally, I could give you credit for a question if you make a good enough argument. So again, I just encourage you to come see me. This question was just kind of a straight definition, but it was one that I guess a term that we didn't use too often. Maybe we didn't use after that. So arrangement of cells. This was in chapter 11. Cube-shaped packets. So a cube is 3D. All right. So that's Sarcina. Tetrads are 4 cells, and I can see how that is a little bit confusing, but I think that was the most common one that people said when they missed it. Okay. Okay, malaria. So which of the following correctly describes part of the life cycle of the causative agent of malaria? It's not a fungus, so you have to read every word means something. So you have to read it carefully, sometimes a multiple choice, oftentimes. The correct answer here is B. Growth and maturation of those parasites in the human red blood cells is what causes illness. That trophozoite stage is actually in the red blood cells. That motile feeding stage, that's when it's doing all the damage. Doesn't harm mosquitoes. And this stage is again in the red blood cells, not in the mosquito. Okay, so that's where it's causing the harm. This was one that I clarified even in class, so I was kind of surprised. Masses of hyphae, so they're really the best answer here. Mycelium, that's a mass of hyphae. It's often underground. It wouldn't have to be underground actually, but like in the terms of a mushroom, the filaments, the hyphae are going to be underground. Mycorrhizae can also be a mass of hyphae, but that's a specific kind of hyphae that's always in a symbiotic relationship with plants. The roots of plants and helping the plants absorb water. Okay, so I would have said something about it's a symbiotic relationship because that's by definition what the mycorrhizae is. All right, so again, that one was another one that was a little bit tricky discerning between those two. I knew that this one would be difficult. We talked about cell growth and what has to happen when a cell grows, specifically when the cell wall gets larger. There's two enzymes, autolysins and transpeptidases, that we kind of defined and talked about. The autolysins are the one that breaks the bonds between the NAM and NAG sugar subunits of peptidoglycan. Okay, so if the autolysins are not functional, if you can't break those bonds so that new sugars can be brought in, then essentially the cell is not going to be able to get any bigger. Okay, so answer E here, the cell wall wouldn't be able to grow. You can't break those bonds. Transpeptidases are enzymes that help form them again. And we talked a little bit more about those because that's what antibiotics act on. The autolysins are important too. Let's see, one more here. Which of the following describes a method of membrane transport only performed by eukaryotic cells? Group translocation is only prokaryotes. That's the one where the substance is modified as it's brought into the cell. Sometimes it's like phosphorylated. Phagocytosis, correct answer here. Remember, prokaryotic cells almost always have a cell wall. So that rigid cell wall cannot endocytose. It cannot extend pseudopods to take in any kind of thing that it needs to transport in. Okay. All right. So if you have questions about those or questions about other things, again, see me afterwards. I'm available after class today. Office hours next week, or maybe we can Zoom tomorrow. Any of those things are available. Okay, so I know that it's been a tough week for a lot of you. There's not a lot of you here today. I know I've seen you rushing from one exam to another, but we do need to go through pathways today. So try to engage today. It will help you in the long run if you don't kind of zone out through some of this. If I'm going too fast, just, you know, can you say that again? Or can you slow down? Don't be afraid to say that. I do want to, last time we tried to lay some of the groundwork a week ago when we were here. And we also kind of got into the beginning of our respiration. We talked about glycolysis. And we also talked about an alternative pathway for glycolysis that some bacteria can do. All right, so I want to start out, oh, here it is, just with a question about that pathway, the Entner-Doudoroff pathway. So take a look at that for a minute, and we're going to review that briefly before we press onward. See if you can decide what's the correct answer there. I already gave some of it away. All right, so see if we can go through these and see why it's wrong or why it's right. Okay, it's an alternative form of glycolysis. It's not just another name for glycolysis. Remember, the other name for glycolysis is Embden-Meyerhoff-Parnas pathway, or sometimes we call it EMP pathway. Okay, so that's not the correct one. It's common to most forms of life. Glycolysis is not Entner-Doudoroff. Only some bacteria can do that. Correct answer actually is that it produces half the ATP as glycolysis. And we'll review that in just a second. Remember, anabolic means biosynthetic. If we're getting some ATP out of it, then it's catabolic, not anabolic. And it's carried out at the same time as glycolysis. No, organisms will only do one or the other. There are a few bacteria that are able to do both depending on what kind of medium that they're in. I think they only do either one or the other. And not, a lot of pathways, an organism can be doing lots of different pathways at once, but not the same for Entner-Doudoroff. All right, so where we left off last time, we were comparing the Entner-Doudoroff with glycolysis. And I know we got to this just right at the end, so I just wanted to review a little bit. Like I mentioned in answer to that question, one ATP goes in, and then we get 2 out. So we're only netting 1 ATP. Okay, so that's different than glycolysis, where we would get 2 ATP. We also get an NADPH, so that's some electrons, some reducing power, but remember the NADPH, those electrons go to power biosynthetic reactions. So I guess you could argue that it does in a way help an anabolic reaction, but not sort of directly. We also get an NADH, and we're going to see where those go today and how those get converted into lots and lots of ATP. Still get 2 pyruvates, so just like glycolysis. And remember, the reason we're talking about this is that this is unique to some species of bacteria. It's a little bit more widespread than we originally thought it was. You don't have to memorize what kind of bacteria do it and which ones don't. I just listed some of them because some of those organisms you'll use in lab this semester if you're in lab. Okay, so there's actually another pathway that's considered an alternative to glycolysis, and that's called the pentose phosphate pathway. Okay, so let's make a note of that. It's an alternative to glycolysis, but organisms can be doing pentose phosphate at the same time as glycolysis, or they might do it at the same time as Entner-Doudoroff. Okay, because you can see here what we're starting with the glucose, 6 carbon. Why do we always start with glucose? That's usually the most readily available energy source to the cell. We'll talk later about how other things can be metabolized, like proteins or lipids, and a little bit about how that might happen. But usually a cell is going to choose to use glucose if that's available. Okay, so when is an organism going to shunt glucose through this pathway? When the cell's needs are biosynthetic. Okay, so it doesn't necessarily need energy, it needs to make cellular structures. Okay, so it wants to have a pathway to kind of power that. All right, so we call this pathway the pentose phosphate pathway. Hopefully that will be obvious here as we move forward, because we get pentose sugars out of it, 5 carbon sugars. It's also known as this hexose monophosphate shunt. I won't call it that. I just wanted to kind of put that in parentheses because I've seen it called that. We're going to call it the pentose phosphate pathway. So if we follow this, and your book really gives like a paragraph to this, and it doesn't even put the pathway in the text anymore. So you're not going to find a lot of background about this. I just kind of want to hit the highlights here. Just like glycolysis, we have an input of energy of ATP at the beginning. And then right away, though, we have a difference from glycolysis. We're getting out some NADPH. Okay, and once again, the NADPH goes to power biosynthetic reactions. It does not get converted into ATP. And we get actually a couple of NADPHs. Oh, so that's written right here, to anabolic reactions. Okay, and then we lose a carbon here to carbon dioxide. We call it a decarboxylation. So now we have a 5-carbon sugar. That 5-carbon sugar, kind of confusing with all different arrows here. It doesn't mean that both of these products get made, because you can't get 2 5-carbon molecules out of 1 5-carbon molecule. So it knows what it needs, and it will either kind of go one way or the other. So it can make one of these 5-carbon sugars. And these are used to synthesize nucleotides. Okay, pentose sugars, 5-carbon sugars. Okay. Let me just write this a little bit. Okay. All right, so key pentose sugars. Used to build nucleic acids. Okay. And again, depending on what the cell needs, it can kind of shuttle down, and you can get a 3-carbon compound here that goes right back into glycolysis if, oh yeah, we need some energy after all. Or we can end up with this erythrose 4-phosphate, which is used to synthesize amino acids. Okay, so erythrose 4-phosphate to synthesize actually aromatic amino acids. So that means amino acids with a ring. Humans do the pentose phosphate pathway as well, but we cannot synthesize aromatic amino acids. That's something that only bacteria can do. Okay. And then again, depending on, kind of what the cell needs or whatnot, some of those carbons can be shunted back over to kind of various steps in glycolysis. Okay, so in terms of what does the cell get out of it when it uses this pathway? Again, it gets electrons that help power biosynthetic reaction, key pentose sugars or erythrose 4-phosphate for nucleic acids or to build amino acids. I would say no ATP directly from this pathway. Your book says that the cell gains 1 ATP, but that's assuming that something ends up, kind of leftovers end up going over to glycolysis. And we call that glycolysis. We don't call that pentose phosphate pathway. So I would take a little bit of issue with that. I would say pentose phosphate directly, no ATP directly. Okay. All right, so kind of a lot going on there in terms of that first step of respiration, either glycolysis, Anaerobic or pentose phosphate pathway. What happens after that in a respiratory pathway? We usually have this kind of transition step between glycolysis and the Krebs cycle, because remember, we end up with pyruvic acid with end of Anaerobic or glycolysis, and that's a 3-carbon compound. Krebs cycle only accepts 2-carbon compounds. So it has to be a little bit of a transition in there. We lose a carbon to carbon dioxide and a decarboxylation. So now we have this 2-carbon acetate. It attaches with coenzyme A in a redox reaction. So we do get a little bit of reducing power here. and that gives us acetyl coenzyme A. All right, so we get 1 per each pyruvate, or you could say we get 2 for each glucose. Okay, so I'll just write that here, 1 NADH per pyruvate, or pyruvic acid, same thing. Or you could say 2 per glucose. Okay, you always have to be clear about how you're counting things. And of course, we get 1 acetyl CoA, again, per pyruvate, 2 per glucose. All right, that coenzyme IA is necessary to attach that 2-carbon compound to the beginning compound in the Krebs cycle. Okay, so moving on to the Krebs cycle. Remember, we got lots of different names for different cycles. We like to give homage to Hans Krebs for elucidating the cycle, but it's also known as the TCA cycle, or tricarboxylic acid cycle, because there are many intermediates in the cycle that have that tricarboxylic acid group on it. We also call it the citric acid cycle, because one of the first intermediates there is citric acid. Okay, so recognize all 3 names, they're all the same thing. So we start out with this 2-carbon compound attaching to oxaloacetic acid. All right, so you have 4-carbon oxaloacetic acid. And now we are, again, with the addition of that 2-carbon acetate, now we have the 6-carbon citric acid. All right, those are the only 2 intermediates I know of this cycle off the top of my head. So don't go, again, memorize the structures here. We're not worrying about what enzymes are involved, just kind of what we start with, what we end with, what we get out of it. Okay, this is all taking place in the cytoplasm for bacteria, for prokaryotic cells. In eukaryotes, it's going to be in the matrix of the mitochondria, I believe. Question? Okay, good. So repeat what we start with. So basically, the cycle is starting out, the acetyl-CoA binds with the oxaloacetic acid, 4-carbon, to give us citric acid. So that's generally where we kind of begin thinking about the cycle. Okay, that answered your question. So thinking about what we get out of this. So I wrote kind of off to the side here, following the carbons. Okay, so citric acid is 6-carbon. Pretty quickly here, we lose a couple of carbon dioxides. So we're back down to 4 carbons. That's interesting because, right, we started out with a 3-carbon pyruvic acid. We lost 1 carbon in that transition step, and now 2 more. So now, like, we've lost all 6 of those carbons that were originally in that glucose. Okay, some organisms can't afford to lose all that carbon to carbon dioxide. So they have kind of a special shunt in the Krebs cycle so that they don't lose that if they need those carbons for, building a cell wall or something like that. But that's kind of interesting. We don't really call carbon dioxide a product of the reaction so much, because it's not necessarily something the cell can use. We get lots and lots of reducing power. So here we've got an NADH, NADH over here, and that other carrier, FADH, we got one of those over here. So we're getting, let's count this per pyruvate. We're getting, and let's count the transition step. You always got to be clear what you're counting and what you're not. So I'm counting that 1 NADH we got in the transition step. So that's 1, 2, 3, 4 NADH. and 1 FADH. So I hope you can read my writing, it's pretty sloppy. Okay, and then if we could count that per glucose too, which we would just double it. Okay, so 8 NADH and 2 FADH2. Okay, again, remember I'm counting that 1 NAD in the NADH in the transition step. So lots and lots of electrons, and we'll see how those get converted into ATP. We're actually only gaining 1 ATP per pyruvate, and that's by substrate level phosphorylation. So remember that term where the phosphate used to add to the ADP to make it ATP comes from some other substrate. And that's not totally clear here what's going on. It's a little bit more obvious in glycolysis where it's ripping a phosphate off of one of those intermediates. Here we initially actually get GTP, but that very quickly gets converted into ATP. I'm not really sure. A biochemist could explain why that is better than I could. And the phosphate here, even though they don't show it on this molecule, is sort of transiently attached to that molecule, I believe. Okay, but we still call this a substrate level phosphorylation. Let's see. So again, we could add on here that we're gaining 3 CO2. Again, that really doesn't do anything for the cell. Okay, and we would just double all those numbers if we were getting our number per glucose. Okay. All right. So that's a sum up of the Krebs cycle. Okay. Another question. Okay, why do I double it for glucose? Because remember, we're getting 2 pyruvates. Okay, we get 2 pyruvates out of each glucose. Okay, so each one of those pyruvates gets changed into acetyl-CoA and goes into the Krebs cycle. Okay, good question. All right, so getting our bearings again, and I don't know if I might not have kept reposting this kind of overview, but remember, we're in respiration. Now we want to know what happens to all these electrons that are coming off, not only the Krebs cycle, but remember we got some from glycolysis as well. What happens to those? They're going to get funneled into some kind of electron transport chain. Okay, so series of redox reactions, they are always going to take place in some kind of membrane. Okay, so for bacteria, that's the plasma membrane. At eukaryotes, it's going to be a mitochondrial membrane, the inner membrane of the mitochondria. I think I wrote that somewhere later, maybe not. Okay, so I'll write that again here. So for bacteria, in the plasma membrane, and eukaryotes, it's that cristae, that inner membrane of the mitochondria. Okay, so 2 different views here to explain everything that's going on in the electron transport chain. Your book gives you 2 different graphics, and one view is a little bit better at showing some things, and the other view is better at others, so it's good to have both. So first of all, this is supposed to be inside of the cell. It's not totally obvious, I think, and this is supposed to be outside. The way I can tell that is because these protons, these are getting pumped out. Whoop, I said that the wrong way. Just seeing if you're awake here. This is outside the cell, and this is inside. Sorry about that. All right. So let's start at the beginning. All right. So here's an NADH, and it's dropping off its hydride ion. Remember, it can carry 2 electrons and a proton, right? The electrons usually travel with the protons, and we don't write them in the equation all the time. Okay, so it's dropping off 2 electrons and it gets oxidized back to NAD. Okay, so that frees up the NAD to go back and pick up electrons at glycolysis or the Krebs cycle. Okay, so I'll come back to this again, but so when the NADH is oxidized, that's oxidative phosphorylation. That's where that name comes from. So all the ATP that we're going to gain here is from oxidative phosphorylation. Not because oxygen is necessarily involved, but because the NADH gets oxidized. Okay, oxidized to NAD. All right, so it drops off its electrons to a series of electron carriers. And those I don't necessarily need you to memorize, or not the order, because that can be different from organism to organism. But you should kind of recognize here, you know, what kinds of things are considered electron carriers. So I wrote that down at the bottom here, flavoproteins. Those are things that contain flavin, which is actually a coenzyme. It can go back and forth fairly easily between an oxidized and reduced state. Cytochromes, so FMN, that's a flavoprotein, flavin mononucleotide. We have several different cytochromes here. There's cytochrome A, cytochrome B, cytochrome C. Okay, those are proteins that have iron in them. Again, can switch back and forth easily between being oxidized and reduced. And then there's this coenzyme Q here is considered a ubiquinone. It's just another protein carrier. All right, so if I mentioned one of these 3 things, cytochromes or ubiquinones or flavoproteins, you should just know that those are electron carriers. Okay. Otherwise, again, don't memorize that. But those electron carriers, some of them are passing electrons. Others of them are also pumping protons outside of the cell. Okay, so that's where I started there, pumping these protons out. If you remember a picture of a dam that I showed you last time, a huge dam with a big reservoir holding it back. Okay, these protons, those are representing the water in the dam that's being held back. Potential energy, because we only let those protons come back through the membrane one little spot. Okay, so that kind of like the dam that gets electricity from the water flowing through the turbines, those protons, when they can flow back into the cell, generate the energy to phosphorylate ADP to ATP. Okay, and that's not really shown very well right here. I'm going to show you at what point that happens in the next slide. Notice here that it is drawn downhill in a diagonal. That's purposely because that's the direction the electrons flow. Okay, like when you put a ball at the top of a hill and it flows down. Well, if you only put it partway up the hill, it would fall down a little bit slower, right? So it has more energy when it's at the top of the hill. All right, so it's kind of, we know that that's the directions the electrons flow. We can measure the electron potential and we kind of understand that. But that second carrier, the FADH2, that actually drops its electrons off at a little different point there. So we estimate that we only get about 2 ATP for each FADH2, and we get about 3 ATP for each NADH. Okay, it's kind of annoying here that they drew the ATPs in here in yellow. That's not actually where the energy is being generated. It just meaning to show, oh, this is about when enough electrons have been passed to equal an ATP. But don't worry about that ATP just yet. Okay, and at the end of all this down here, okay, there's going to be some final electron acceptor. right? The next picture kind of elucidate that a little bit easier. But know that this could be oxygen. If this is aerobic respiration, this is going to be oxygen. It can be some other kind of acceptor. Usually something that's an inorganic ion. All right, that here, we'll come back to that. If it's anaerobic respiration, it's going to be some inorganic ion like nitrate or sulfate. Okay, but we'll talk more about that later. Okay, don't just assume that it has to be oxygen. Okay, the other picture in your book, oh, I knew it talked somewhere about where this was taking place. So it shows you a little bit better like where these processes are actually taking place. Here it's dropping off the electrons, you know, with NADH and FADH a little bit further down. And you can see all these protons that are outside of the cell. Okay, that's building up a gradient. Okay, they're going to flow from high concentration to low concentration. And where that's going to happen is an enzyme there in the membrane called ATP synthase. Okay, this is why it's extremely important that the membrane be intact. Okay, if there's something that's poking holes in the membrane, whether it's an antibiotic or some aspect of our immune system, talk about complement proteins that do that. If that membrane is not intact, then those protons can just flow in anywhere, and then we don't get the ATP from that. Plus, things are going to be kind of get lost from the cell. But in terms of energy generation, that's really bad if there's holes in the membrane. Okay, so say oxygen is the final electron acceptor here. It's actually, oxygen is split so that it can accept those electrons and we end up with water. Now the oxygen is not always split correctly. So remember we talked about toxic forms of oxygen. Okay, this is where we can get the superoxide. Okay, and it's estimated that about 5% of the oxygen that we breathe ends up as superoxide. Okay, so this happens pretty commonly where the oxygen doesn't split correctly, and we have this toxic oxygen product that, remember, has to be detoxified. Cells make superoxide dismutase to detoxify that. Okay, if cells cannot make that, then they can't respire aerobically. Okay, or and in addition, we get a little bit of some of these electrons that are being, you know, passed from carrier to carrier. Some of them kind of leak through the membrane now and then, and you know, can form inappropriately with oxygen. So again, we get some. That's where some of the toxic oxygen forms come from that cells have to be able to detoxify. Okay, let me see. I had a lot to talk about there. Let's see, I forgot to say anything. Okay, and remember I said that oxidative phosphorylation. So even if oxygen isn't the final electron acceptor, okay, because the NADH is being oxidized, that's where we get the term the ATP generation is by oxidative phosphorylation. Okay, questions here before we move on. A few terms here that you hear in conjunction with the cell membrane: proton motive force and chemiosmosis. Both those terms are really closely related, but I want to kind of parse that out a little bit. Okay, the whole reason that ATP gets generated is due to that proton motive force. So that refers to the protons that are building up outside of the cell in high concentration, and then again they flow down their concentration gradient from high to low. Okay, so that potential energy is known as the proton motive force. Then when they actually diffuse into the cell, we say that ATP is generated by chemiosmosis. Okay, so you know what osmosis is. That's a movement of water from high concentration to low concentration. This describes the movement of ions from a high concentration to low concentration. Okay, so due to the proton motive force, we get chemiosmosis, which actually powers the phosphorylation of ADP to ATP. Okay, so just hopefully clarify those terms a little bit. Question? Yeah, so repeat proton motive force. So that just refers to when the electrons are passed down the electron transport chain, the protons that are associated with them get pumped outside of the cell. So when there's lots of protons, more outside of the cell than inside of the cell, that's the proton motive force. That's like the water in the reservoir that the dam is holding back. That's lots of potential energy, okay, because there's so much of it. Okay, good. All right, so if we think about numbers for a minute, aerobic respiration. So, okay, that may have had my overview. We're thinking about, especially about ATP production, but kind of sum up in general what we're getting from all three parts of aerobic respiration. Assume a cell's using glycolysis, Krebs cycle, and electron transport chain. Like I said, with the NADH, we kind of assume that approximately 3 ATPs are produced per NADH. Okay, so that gives us 30 ATP from. So that's our, when I say it's all about the electrons, it's all those electrons that are getting converted into ATP, and about 2 ATP per FAD. So that gives us 4 ATP. Okay, so if you add all that up together, theoretically, for prokaryotes, we should be able to get 38 ATP out of aerobic respiration. And for eukaryotes, it's a little bit different. It's considered theoretically to be 36 ATP because 2 ATP are spent in transferring these NADHs from the cytoplasm where glycolysis is taking place into the mitochondria. Okay. Now those numbers are theoretical. Does this ever get reached? Probably not for bacteria because they utilize some of that proton motive force for other things. They actually power their flagella with that proton motive force. So not all those protons are going to be flowing back into the cell and being converted into ATP. It's being actually used to power the flagella. So those are the theoretical numbers that you kind of always hear. What if the cell is using something other than glucose? We're always talking about glucose, but we know that most cells are flexible and they can use certainly other sugars. Other sugars are typically, as long as the cell has the enzymes to kind of break the bonds, if it's a larger sugar, those are usually shunted in or maybe turned into glucose. So that's very similar. Proteins and lipids are a little different story though. And I like to call attention to this because if you're in lab, it didn't match up quite right this year. I don't know what order I changed. But a couple weeks in lab, you're going to be testing for a couple of different proteases that some bacteria can produce. Okay, so if a bacteria is going to be able to use a protein for energy, remember, they can't just engulf it. They have to break it down outside of the cell into smaller pieces that can then more easily transfer through the cell membrane. So they have to secrete some kind of protease to chop up that protein, that polypeptide, into its amino acids. And then the amino acids are usually transferred in this can happen in several different ways, but one of the ways is that you have a deamination reaction. So that means you're chopping off the amino group from the amino acid. And you're going to be testing for an enzyme in lab that's a deaminase. Okay, organisms that can make that deaminase are able to use that particular amino acid. Okay, so when we have a deamination, we get this NH2, that amino acid amino group, that often gets converted into NH3, which is ammonia. Okay, what do you know about the pH of ammonia? Okay, alkaline. All right, so the so what here is when organisms catabolize proteins, you're going to have alkaline end products. Okay, that's sort of important to keep in mind. When sugars are metabolized, you're almost always going to get acidic end products. So we do various tests with pH indicators in lab, and depending on whether it's an alkaline pH or acidic, that gives us really useful information. And then, so what's left over here? You got a 2-carbon compound. And we just talked about how, oh, it's 2 carbon compounds that are able to fit into the Krebs cycle. Okay, so we know we can get lots of reducing power out of that, even though kind of glycolysis was bypassed a little bit. That can get shunted right into the Krebs cycle and get lots of energy from that. Okay. A little bit about lipid metabolism. I just want to touch the surface here. Let your biochem professor go into this in more detail. But again, we talk about this in lab. We test for organisms whether they can make an enzyme to break down lipids. Okay, we just call it lipase. There's probably different kinds of lipases. But lipids are composed of fatty acids and glycerol. Okay, so basically lipases just cut that bond. So like right here, the 3-carbon glycerol and the fatty acids. Okay, so we call that's a hydrolysis. It breaks the glycerol off from the fatty acid chains. That's lipase that does that. First of all, what happens to that glycerol? It's a 3-carbon compound. We enter some ATP is inputted, and then we come up with this dihydroxyacetone phosphate. That was the molecule that when in the splitting step of glycolysis, I said, well, this is not symmetrical. We get this DHAP that usually is converted right into glyceraldehyde 3-phosphate, I think. But that's, anyway, that's just one of the steps in glycolysis. So that can go right into glycolysis and get some energy there. But the real energy trove is from those fatty acids. Long chain carbon chains through a process called beta oxidation. Those long chain fatty acids are cut off 2 at a time, okay, with the help of coenzyme A. So we get some electrons out of this just kind of adding the coenzyme A, but you can see here we get stepwise process of these 2-carbon compounds come off, and those go straight into the Krebs cycle. Okay, so think how many of these 2-carbon compounds we get just from one fatty acid chain. So lots and lots of ATP. Again, let me just write this a little bit bigger. Beta oxidation, it's just kind of the stepwise breakdown of these fatty acid chains. Again, lots of ATP will eventually result from that because we've seen where all those electrons go from the Krebs cycle, okay, into electron transfer chain. Okay. Neat. How are we doing? Okay. All right. So your book really doesn't spend very much time talking about specifically anaerobic respiration, which seems kind of odd to me because that's something that only bacteria do, right? Again, this is only bacteria. We never do this. We don't have the correct enzymes to use anything other than oxygen as a final electron acceptor. It's true that a little fermentation goes on in our muscles, but that's a different process, and we'll talk about that in a minute. So this is just when there's some electron acceptor other than oxygen. Okay, and I mentioned it before, it's usually some kind of inorganic ion. So remember, when I say inorganic, that means it doesn't have carbon-hydrogen bonds. Okay, so what's some examples of inorganic ions? Nitrate. Okay, and I mentioned that one first because this is another one you're going to test for the enzyme in lab that enables the organism to use nitrate as a final electron acceptor. It's called nitrate reductase. Okay, so nitrate accepts those electrons and gets reduced to nitrite. Okay, if a molecule loses an oxygen, that's a reduction. Okay, I don't have all the electrons. If I wrote this out, NO3 plus 2 protons plus 2 electrons yields nitrite plus water. Okay, 1, 2, yeah. All right, so the full reaction. All right, so I'll come back to this page in a minute, but I don't think I posted this, but I just wanted to show you organisms that can make this enzyme nitrate reductase. Okay, so that's this enzyme right here. It's nestled right up with a cytochrome right in the electron transport chain. Okay, if the organism has that enzyme, then they can use nitrate to take those electrons. Okay. So then the organism can live in places where there's no access to oxygen. Okay. What other electron acceptors? Sulfates. Okay, that's another one. Sometimes I don't have the reaction in my head here, but hydrogen sulfide is the end product. I should write that out. Okay, hydrogen sulfide is another thing that we'll test for in lab later on. That's the so supposed to be the really stinky smell, like if you've ever been to Yellowstone, the kind of sulfury smell around some of the geysers. Okay, that's bacteria producing hydrogen sulfide as a result of respiring anaerobically. Let's see, what else? Sometimes iron is the final electron acceptor. Carbonate. Organisms that use carbonate as a final electron acceptor. Actually, again, I don't have the whole reaction there, but end up with CH4, that's methane. A really very potent greenhouse gas. So let me show you, come back to that. This picture. So I didn't post this either. But this is why, if you've ever been to a landfill, they have kind of pipes sticking up. You know, they kind of bury all the trash all the time, but then they keep these pipes sticking up. That's to try to aerate it a little bit, because you get buildup of microorganisms that are respiring anaerobically and making methane, which if you get that build up too much can actually explode. So you don't want that to happen. The landfill just south of town here in Grand Rapids actually collects some of that methane and uses it to power some of their electricity needs there at the landfill. So they're doing this on a really small scale. It seems like a great idea to take advantage of something that bacteria are just doing anyway. We don't have this, so it's really economically feasible. Like, it costs so much to, you know, outfit the plant to collect the methane that it just doesn't pay itself off in a kind of feasible amount of time. But it's kind of a cool application. Okay, so let's see. So again, what I'm talking about here when I say anaerobic respiration, okay, I'm referring to this green pathway organisms that use something other than oxygen here, they use glycolysis, kind of a little modified Krebs cycle. It's not exactly the same, and electron transport chains. Okay, so they're still harvesting lots of ATP, but actually not quite as much. Okay, so this is the point that I'm trying to make from the slide, and I'll talk through this in a minute. But I'll just write this here: organisms that respire anaerobically don't get as much ATP as aerobic respiration. Okay, so if they use something other than oxygen, like nitrate or sulfate, they're not going to get as much ATP out of the process. Okay, but they can live in places where there's no oxygen. So that's an advantage. Okay, so I could just tell you this, that they're not going to get as much ATP, but I want to try to show you why. If you understand a little bit why, then you're more likely to kind of understand that concept. So we can actually test, I mentioned this before, we can actually put electrodes into cells and understand kind of the flow of the electrons, or we could say the electron potentials. Okay, so when we have done that, that helps us understand like where the different carriers are, the exact order of them, and we can actually measure how likely a molecule is to give up an electron. Okay, so we measure this here. This is called the E naught prime. So the little circle there, we pronounce that naught, and there's a prime. And that's just a measure of how easily a molecule gives up electrons. Okay, so measure how easy a molecule gives up electrons. Okay, so for instance, if we look at this pair, NAD, NADH, okay, that's pretty high on the electron tower. So the NADH is quite fairly negative here, and so that means that it's easily going to give up its electrons. Okay, very glad to do it. Okay, if we look at down at the bottom here, if we look at oxygen, well, and whoops, don't worry about that calculation. Down here at oxygen, it has a very positive E naught prime. So that means it wants to gain electrons. Okay, so gains electrons. Okay, so molecules at the top give them up, molecules at the bottom want to get them. Okay, so the distance from where the electron gives it up and when it's gained, when that distance is greater, then that converts into more ATP. And you could do that calculation to figure that out. I'll leave that for your, again, biochem professors. But you can see when we have the NADH drop off its electrons, that's the furthest distance here from oxygen. Other things, if other things are used as a final electron acceptor, like here, sulfate or nitrate, it's just not as long of a distance between what's between the acceptor and when it's dropped off. So here we've just got a very short distance with sulfate. Here a little bit longer. If you use iron, that's almost as far down as what oxygen is, but not quite. So again, what I'm trying to say is you're always going to get less ATP when a cell respires anaerobically than if it uses oxygen. And sometimes organisms can use both. Okay, when would an organism respire anaerobically? Well, if they are considered a facultative anaerobe. Remember, we talked about oxygen requirements a little bit. I'm going to talk about that more in lab next week when you do thioglycolate tubes. But remember, a facultative organism is one that's flexible in its metabolism. So a facultative anaerobe can respire aerobically, but it can also do some kind of anaerobic respiration or maybe fermentation. Talk about that in a minute. An aerotolerant anaerobe and a strict anaerobe would all be able to most likely respire anaerobically. Okay, so what's the difference? We're getting there, we're almost through. What's the difference between anaerobic respiration and fermentation? Those are two processes that a lot of times are just kind of grouped together because neither one of them require oxygen. Okay, so neither require oxygen, but they're actually very different processes. Okay, the difference is really the fate of pyruvate. Okay, what happens to pyruvate makes all the difference. So if I go back to our overview here, we've got the pyruvic acid right here. If there is some electron acceptor available, whether it's nitrate or oxygen or whatever, respiration will take place. If it's not, then it has no choice but to enter into some fermentative pathway. Okay, so I've got the dictionary definition here, but typically the final electron acceptor in a fermentative pathway is some organic molecule. Usually that means some kind of molecule that's already in the cell. Okay, it doesn't need to get it from outside of the cell. So very often pyruvate just acts as an electron acceptor. Okay, so the whole point there with the fermentative pathway is really just to catch these electrons that are given off of glycolysis with NADH so that the NAD can cycle back and keep glycolysis moving. Because if that NADH doesn't have anywhere to drop off its electrons, then it can't cycle back and glycolysis even will stop. Okay, so usually we get no ATP from some kind of fermentation pathway. Only the cell's only ATP would come from glycolysis. Okay, but at least they get that ATP. Well, that's not always true. We're going to look at some different pathways. Sometimes the cell gets a little bit of ATP, but most often it's just trying to keep glycolysis going. So it has to, again, oxidize that NADH back to NAD to keep glycolysis going. All right, so your book gives you one example of a couple examples of fermentative pathways. So it actually is two different pathways. It kind of makes it look like it's one. The most common, the simplest fermentative pathway is a lactic acid fermentation. So where that pyruvic acid that we end up with glycolysis, there's just one enzyme involved there that changes changes this oxygen pyruvate. That's where it accepts that proton and that electron and changes it into lactic acid. Okay, so there's still a lot of energy left in that lactic acid, but without some other kind of electron acceptor, it really can't get any more energy out of that. But again, at least it keeps glycolysis going. That's the simplest fermentation pathway. One change, there's not any gas produced there, no ATP produced. So usually no additional ATP. And I'll just say usually. Okay. And for bacteria, typically you're going to have some kind of acidic end product. lactic acid. Okay, that's going to be really, really acidic. So again, we can use a pH indicator to test and see what the medium is like. And if acidic end products results, then we know there's been some fermentation taking place, because we're talking really acidic, you know, like 5, pretty easily detected. Most of the time, bacteria, like I said, will have acidic end products. Yeast, on the other hand, can do a different kind of fermentation. Okay, that's this pathway over here. They get a little gas produced there and end up with alcohol. All right, so that's not common for bacteria to do that, but yeast will often do that fermentative pathway. Just kind of depends on organisms. Some organisms will always do one pathway, and other organisms have different pathways. It depends on the enzymes that they can make as to what kind of pathway that they do. Okay, but again, I said this before, but the purpose here is just to keep glycolysis running by oxidizing NADH back to NAD. That's really the purpose of a fermentation. If you took a good microbial physiology course, you'd talk a lot more about this and get into lots and lots of pathways. I wanted to show you this picture. I did not post it because I didn't want to scare anybody, but there's lots of different fermentation pathways that bacteria do. Most of them will produce some kind of a gas as a byproduct, but they really can produce all kinds of really cool stuff. One of my favorite fermentations is these propionobacteria that actually make propionates. That's the distinctive flavor you get in Swiss cheese, right? Carbon dioxide is given off. That's the holes in Swiss cheese. So it's cool when, you know, bacteria do good stuff for us. In lab, later on, we will test to see if an organism can do a certain fermentative pathway. You'll test to see if it can do this 2,3-butanediol fermentation. That helps us identify organisms when we understand the enzymes that they can make to do a particular fermentation. So a whole group can do that 2,3-butanediol. Another group does what we call a mixed acid fermentation, where acetate is one of the end products. The Clostridia bacteria. So if you were in lab, you stained some Clostridium. Remember, they're spore formers. They can do a lot of really wild stuff. This butyrate, that's a kind of an intermediate in one of the fermentation pathways, that's the smell that's associated with C. diff diarrhea. It's supposed to be one of like the nastiest smelling things that there is. Apparently, some nurses have said that they can identify if an organism has C. diff diarrhea just by the smell of the diarrhea. Call it the whiff of the diff. Right, so nurses, this is what you're getting into. Also, one of the kind of bad smells in vomit too. So somebody said to me once, I just don't find this class that interesting. And I said, wait, the whole like deodorant industry is based on bacterial fermentations. Like, this is interesting stuff. It's very applicable to what we do all the time. So, okay, so anyway, fermentation. Next week in lab, I got this didn't line up just right, but you'll be inoculating fermentation tubes. So you'll have certain sugars in the tube, and then you look to see if you get a pH change, an acidic pH change. And if you do, then, oh, I know that that organism can metabolize that sugar. It's got the enzymes to break the bonds. We put a little tube inside of it to catch the gas. So there's a little gas bubble in that tube. It's hard to see it, but just as a further way to characterize, well, does it use a fermentative pathway where gas is produced, or does it use some pathway where there's no gas in there? So just kind of on the horizon if you're in lab. And I wanted to show you this picture from your book, not to memorize at all, just to show you, we get very useful products from bacterial fermentations. I mentioned Swiss cheese. There's lots of other kinds of cheeses, soy sauce, yogurt. Okay, all possible because we have bacteria doing fermentations, alcoholic beverages, even things that you don't think about being like organic, like nail polish remover, rubbing alcohol, vinegar, acetic acid. You know, all that's produced by bacteria. So again, it can work for us and not just against us. And I also wanted to post just this table. A lot of you have been asking me, how can I study better? Okay, as much as you can when you're studying, you need to be active. So you're not just reading over things. I mean, flashcards is one way to be active. I think when you're studying a topic, you need to be quizzing yourself as you go as much as you can. So here's an example. Here's a table. Cover up part of that table and see if you know the answer to that. So this is just kind of basic differences between anaerobic respiration, aerobic respiration, and fermentation. We didn't really talk too much about the output here. That's really variable, depending on what pathway the organism is using, or what is the electron acceptor. But again, this is just one way, you know, cover up, quiz yourself as you're looking over things, okay? I'm gonna let you go 5 minutes early today because that's a good time to stop. Hope you have a good weekend.